Thanks very much. Thanks, Vola, Jennifer, and Judith, and Dimitri, and Joanna, for this initiative and for the honor of being the second speaker. So I hope I'll keep the level as high as you did, said it last week, last month. Sorry. So I wanted to talk about the work that we do in the lab. There's some branches, but the main focus of the lab is in neutrophils. Our little friend here. I'll keep this a pointer. I don't know if you can see the pointer, but I'll be using it rather than the laser for the movies. So yeah, we're interested in many aspects of neutrophil biology, and I call it non-canonical. There was some funny Twitter coming up, why non-canonical? And I guess non-canonical is a good word to say anything that is unexpected. So there you go. We're finding a lot of surprises and away from the mainstream dogma, I guess. And that's a way to catch a bit the attention. Non-canonical means different things for each person, but here you go, non-canonical. So we have a little dude here. The neutrophil has been known for, of course, for a very long time, since basically microscopes were there with enough resolution. And yeah, we know a lot about it, or at least about some aspects, but the non-canonical refers to the many other things that these cells are probably doing. I will discuss some of these things that we found and point to some of the directions where we're going now. So you have a sense of the interest, and you know, for those of you who are not yet a neutrophil fan, hopefully get you a little bit interested that in physiology, this I think are one of the most exciting cells today. So every cell is, of course, but these are at least closer to my heart. So I will discuss some of the work that is already published, and I've discussed in other forums. So I will go relatively quickly through it. Aspects of the circadian biology of neutrophils, aspects about heterogeneity that I just mentioned in the first slide, and some work that is unpublished and that I would love to get some of your feedback before we submit this. So yeah, I will discuss this at the end. So just to start with the canonical neutrophil, you know, as a counterpoint, and of course, one of the most important functions, if not the most important function of this cell, is to keep us healthy and free of microbes. And this is a classical movie from David Rogers a long time ago, looking at the cell chasing with a lot of focus on these bacteria. And this is basically what they do in the body. They just move around, they patrol tissues, the bloodstream, and they make sure that we're clean of it, of pathogens. And we know this in part because if neutrophils are dysfunctional or in neutropenic people or animal models, very often they have a poor quality of life and they succumb to infection. So this is, of course, a primary function. Without this, all other functions probably are not as important, or maybe they're just as important, but this really is primary. If we're not healthy, we don't know what else it is that they're doing. And this is something that's probably keep us away from other truly fascinating aspects of the physiology, biology of these cells. So few words about circadian biology, because I think it's thought-provoking to give this spin to the biology of disease, particularly because, as you know, they're short-lived, so the biology, even their lifespan, adjusts quite well to the circadian oscillations that we all know a little bit about, but turn out to be much more complicated when you go into it. And that really is, I'm not a circadian molecular biologist. I love the physiology, but there's a lot of regulation of these problems. So why circadian oscillation are interesting at all for immunologists or for physiologists in general? And one way to put it is that the place where we live is a changing environment. So this is basically this ball that turns around, this very hot ball. So the conditions in the surface that faces the sun are different from the other surface. This, of course, what organisms do is optimize the resources and adapt to the environment. Without that, species fail and disappear. So one adaptation that requires a 24-hour rhythm, in the particular case of our planet that takes this long to do a full spin, is to adapt to these conditions in temperature, access to food, pathogens, predators that will chase us, food that we chase. So how do we optimize our physiological resources? And one way to do this is by programming specific aspects of our physiology to these known variations to predict what's coming. So a basic aspect of circadian oscillations is predictability. So if we predict these changes, then the body over evolution adapts to these. And in the case of immunology, it becomes just as relevant. Again, I mentioned that there's the threat of pathogens that would be more likely to infect us when we are active during daytime, and rather when we're a nighttime resting, that would be more of a healing phase or repairing phase. So this is a little bit the intention, the evolutionary intention of this, that is difficult to demonstrate, but that's the rationale for most of us studying circadian biology. And you can understand or visualize circadian biology probably in your favorite physiological context or topic of study. It doesn't matter if you study one organ or another, or you just study inflammation, healthy conditions. Almost in any situation, you would find strong variations, in some cases very strong variations in gene expression, migration of the cells, the presence of metabolites, no matter what. But we revised a little bit of this earlier this year, made the point that this axis, the circadian regulation, that for the most part originates by perception of light or intake of food. So these environmental cues elicit a number of changes that have an impact on the specific tissue that you might be interested in, either pathogenic or just physiological changes. And a lot of these entail the function of a number of cells in between. Many of these are immune cells, but what many people have realized is that they don't work alone. They do so in concerted action with other different cell types, what we call the structural cells. Probably basically every cell type would probably be part of these circuits, but we envision this as these structural regulator cells and immune cells, which are usually the effector cells, but in some cases they're regulators themselves of downstream physiological processes. So this crosstalk is what we wanted to emphasize, the different circuits depending on the specific process that would go from this central regulation to a specific cell type that in turn regulates another cell type and has an output on the specific tissue. So these circuits are quite prevalent when you start looking in detail in your physiological context. So circadian biology and circadian immunology is important, and it's a way to look at your specific problem. In the case of neutrophils, a number of years ago, we got interested in this topic. The simplest or most obvious thing that drew us to be interested in circadian physiology of neutrophils was that when we look into the circulation, so the easiest condition, you just take blood in animals where you haven't done anything except you wait 4 hours every between each time that you take blood, and then you analyze it by flow cytometry. 2 parameters only, and where you can see just by looking at the expression of L-selectin CXCR4, the appearance of specific population that is low for L-selectin, high for CXCR4, and this shift is very predictable. It occurs every day with this population highest at noon and then disappearing at midnight. So these are Zeigiver times, basically time after the lights are turned on in your specific facility. And we can draw curves that very much adjust to circadian patterns when you look at this. And I think it's kind of obvious that this follows a cyclic period. The next day, the animals would have the same pattern, basically. So these, with a number of experiments over a number of years, so this took quite a while, allowed us to build what is probably a preliminary design of what we call the neutrophil clock. That is composed of a number of molecules. The ones that we have identified, there are many genes and molecules that have circadian oscillations, also some of them in the neutrophil, but this clock, at least key regulators that we found are BMAL1 that regulates expression of this chemokine, We found that this acts in a cell intrinsic way. We don't know how, but it doesn't affect other cells. It works cell intrinsically, hits this receptor and triggers this. We call it aging because it accumulates over time during one day. But it could be also some sort of activation, physiological activation, not the typical way you hit the cell with a very strong stimulus. This is just a physiological low-grade activation, if you want to put it this way. So this would be the activating hand of this clock. And then we found also the CXCR4 acted as a negative regulator of this process, probably by interfering with CXCR2 signaling. And altogether, this negative and this positive hand make up this clock. One experiment by which we know that the clock regulates this specific aspect of the physiology of neutrophils, because if we look into wild-type mouse, we would find, the description of this pattern that I showed here, an increase in the number of what we call H neutrophils at noon, then they go down and disappear at nighttime. Next day they will come up. If you eliminate BMAL1 only neutrophils, in a mouse, then you would find neutrophils that are unable to engage into this phenotypic change. So they remain constantly low. If you build the animal that's the opposite, it lacks the negative regulator CXCR4 only in neutrophils, then you find the opposite. These cells are in this activated age-like state permanently during daytime. So this is how this reflects into this particular aspect of the physiology of neutrophils. But of course, this is just expression of a couple of genes. So we did a lot of transcriptomic analysis and a lot of experiments that allowed us to predict. So we'll just go quickly through this. This has been published. Allow us to predict that there would be changes in the migratory capacity of neutrophils. So we made an artificial system in which we would have in the same mouse fresh light, which would be night-like neutrophils, and daytime day-like neutrophils at the same time in vessels in about 50-50 ratios. And then we look at whether the cells would behave differently in the context of inflammation, in this case with TNF-alpha. So, we look at the rolling phase, and you will see that even if they're 50-50 in the circulation, you'll find that many more of the green cells, which are the night-like neutrophils, engage into rolling with the vasculature, with inflamed vasculature. These in turn results in more adherent cells after a few hours of the green cells attaching to the vasculature. So the night-like attach better, roll better, attach better, and eventually many more of the green cells extravasate out to the site of inflammation. So this really has a physiological consequence in how the cells migrate to the target area in the context of inflammation. So is this important? And I don't know if you can see the title here because I cannot remove this thing. But anyway, is this important? And we challenge this idea that this clock intrinsically in neutrophils is important to regulate their activity in meaningful ways, so during immune reactions. So we tested, and this is based on this idea that one of the aspects is that they migrate differentially to sites of inflammation, as shown here. This would be the night-like neutrophil, and this is the day neutrophil. So one experiment was to challenge animals with a fungal infection, and in the condition of a wild-type animal, you would find that the nighttime neutrophils, sorry, this came out weird, I don't know why. But anyway, the nighttime neutrophils are more protected than the daytime neutrophils when you hit the animals with the pathogen. So you have this diurnal differential response. Is this dependent on neutrophils? So now we use the neutrophil-specific BMAL mutants, and what we find is that the animals are still immune competent, but this differential response that we found in the wild-type conditions is gone. So this change, this small enhancement in immune responses is what is regulated by BMAL, and we think that this makes a difference when you encounter pathogens, especially at early phases. Your normal conditions, not when you get an enormous wound, but when you need to clean up your system from residual pathogens. So this is just an interpretation of how this would be beneficial to the organism. Now, another setting is when we induce external inflammation. So this is another very important aspect in neutrophil physiology. They are protective, but they're also very damaging, because in some instances they cannot discriminate between a dead cell from the host, and elicit and exaggerate responses that extends the injury to the organ. So this is a big problem in cardiovascular diseases. So we reproduced one of them that we use in the lab. We reproduced myocardial infarction by transient ischemia, and then we look for the amount of dead tissue in the animal. So if you do this experiment in a wild type animal, a little bit like happened here, you get a very strong differential response. In this case, with a stronger or bigger myocardial death when you do it at daytime, at noon, compared to when you do it in the evening at 8 p.m. This would be the time, and this is just images showing the infarcted area, the dead myocardium. Now, if you repeat the experiment with the BML mutants, much like we did here, what we found is that this difference is gone. So you go somewhere in the middle. It's not that you're fully protected or fully compromised, but the differential response is low. So the idea that we extract from these experiments is that there's a lot of decisions that are made in a cell-intrinsic manner by neutrophils, by having this molecular regulator or sensing of time that decides how strong their participation in an immune response or an inflammatory response would be. And this is, I think, important because we thought that in many ways the function of neutrophils was just driven by the environment, you know, just come here and do what you do. In this case, what we found is that there's a lot of regulation in the activation of and the activity of neutrophils that is cell-intrinsic. And this is a little bit how we put this together, this idea of this physiological aging, these changes in the transcriptional profile and functional profile of neutrophils between nighttime and daytime. This induces differential responses, for example, to an infection. So this diurnal defense with change, but at the same time, we find a differential response to a sterile injury, to host injury that does not involve pathogens, that we find it to go in anti-phase. Now, when you go artificially into this system where you are always in a night-like state, what would be the benefit of this? So the benefit would be that you are more protected. That's what we found. I'm showing the experiments of this published. You will be more protected against a sterile injury, for example, in the model of myocardial infarction, but at the same time, you lose this extra kick in nocturnal defense against pathogens, and this might make a difference in the wild. Now, if you do the opposite and you go into a constitutively age activated like state, this is great. These animals, we found they're very much protected against bacterial, against fungal infection. But in contrast, when we induce vascular injury or ischemia in these mice, they tend to die much more readily than the other animals. So really, this looks like the system is built as a compromise. So you focus you strengthen the response to specific times of the days, probably when you're going to be more exposed to pathogens. And then you alleviate that response, probably as a way to protect the host from the toxic activity of neutrophils. So, okay, this is a lot of what I showed is function and transcriptional profiles that gave us hints. But neutrophils work a lot with proteins, by suffering proteins that they store in the granules, which are synthesized during their maturation in the bone marrow. So a lot of what they do, they're not transcriptionally very active, but they store a lot of proteins that they can release when they're challenged. And this has been the dogma for many years. You know, there's a lot of these kind of proteins are just toxic to anything. So you want to keep them inside the cell and only expose them when you are facing, for example, a microbe, a bacteria or fungus. So that was the idea. But anyway, we wanted to see if there was circadian patterns in the proteomic profile of the cells, and we designed this experiment to get night-like neutrophils and day-like neutrophils in large amounts, so we could perform transcriptional profiling with good depth. And what we found, surprisingly, I have to say, is that there was many more proteins in nighttime neutrophils than daytime neutrophils, and particularly when we focus on proteins that were storing granules, those proteins that are important for immune defense and that are toxic, there were also more at nighttime than at daytime. So a simple interpretation that then we could validate by looking at the cells is that these cells and neutrophils, they granulate spontaneously. So part of this aging program, this natural program that takes place during 24 hours in the body until the spontaneous degranulation. These animals, in the experiments that we did in this study, were not challenged with anything. So they were just the animals that are sitting in SPF, not challenged with, you know, as clean as probably they can be. And yet they were degranulating. They were losing granular proteins that were in a very predictable way. Global global proteins from many compartments, but particularly those from granules. And this did not go apparently against the health of the animal, probably because they're released gradually into the serum, which might have efficient inhibitors for some of these more toxic proteins. Now, another interesting aspects when you think about granular proteins that they're important for the this phenomenon of netosis, the release of nets, which is the condensation and ejection of the DNA of the chromatin out of the cell as a way, this is what is predicted, to have a larger area of action and trap microbes and kill them, because they contain proteins that are highly toxic, including histones and proteins that were originally contained in the granules of the cell. So these are toxic nets, if you will, and this apparently should be efficient in containing infections, but they're also toxic because, as I mentioned, they contain a lot of toxic proteins. So they have been involved in many types of sterile injury for myocardial infarction and stroke. And more noticeably, recently, there's a lot of interest in whether this contributes to the lung toxicity in patients with COVID. So what Jose Maria did in this series of experiments is propose that, if you're losing proteins that are involved in netosis, then the same thing that you have a differential proteome between nighttime and daytime, you will have a differential production of these toxic nets. So he did an experiment where he induced acute injury in the lungs in this model of trial, transfusion-related acute lung injury, and looked for the formation of nets in this very ingenious way that he found. So this is the lung of a living animal. We challenge and we induce acute lung injury, and we look for the formation of nets by looking for the extrusion of DNA, which is labeled in red, just simply in propylene iodide, and then neutrophils labeled in blue, as I mentioned, and look at events where these blue cells are spitting red material. This is one example of this. There are a few examples. This is done at daytime, right, when there's the lowest amount of these granular proteins. So we predicted low netosis, and this is what we found. Now we repeat the experiments at nighttime, and we look for the same thing. And what we found is that there's many more of these extrusion of nets out into the circulation, suggesting that the same thing, or what we predicted from the granular content, and translates into this particular form of toxicity of the cell. So this is a quantification of this production of nets. We know they're nets because they're sensitive to chloramidine treatment. There's more at nighttime than at daytime. This translates when we look into the consequences for the lung in more edema, which is measured here by computer tomography, looking in red for the accumulation of water in the lungs of these animals. Before acute lung injury and 21 minutes later, you see this increased accumulation of water, which is stronger if you look at compare the signal at nighttime, and this is what is quantitated here, so quantified here. So there's more lung injury if you do the experiment at nighttime than at daytime, and this translates into less survival of the animals when you do the experiment at nighttime compared to to daytime. And this again is reverted by chloramidine, suggesting that this is net mediated. But this is the whole animal, so we don't know what neutrophils are doing here. So we repeated the experiment now with our clock, and knowing the molecules that are important for this, we generated our daytime and nighttime like specific neutrophil-specific mutants, the BMAL mutant, the CXF4 mutant that I introduced before, and we saw that this release of DNA in vivo from this imaging the lung, it's basically recapitulated, and the BMAL mutants behave like the nighttime neutrophils, the CXF4 mutants behave like daytime neutrophils. And as suspected, the BMAL mutants develop more edema when we induce acute lung injury, and this is shown here, whereas the CXF4 mutants are basically protected from this, and they're, I'm not showing this here, but they're basically a granular, so they lost a lot of they don't have a lot of the granules, which is consistent with what we found, and this also is consistent with the poorer survival of the nighttime BMAL mutant animals when you challenge with acute lung injury. So the idea that we extract from this, just to summarize a lot what we've done, is that this canonical one-line biology or physiology of neutrophils that are produced in the bone marrow, going to the circulation, and they're important to contain infections, but they also participate in inflammatory reactions, including autoimmunity. So this is the canonical view, and this transition that we put here is a little bit to represent this circadian change, and possibly in phenotype, but also in function. So what we find now, and what we wanted to look next, is that the biology of neutrophils in the circulation is not unidimensional, but it's probably multidimensional. And one aspect that we were interested in is in the capacity to invade tissues, to go into tissues, and what is the physiology of the cells once they go into the tissues naturally at the end of the lifetime. Again, no inflammation here. We were probably one of the few labs doing neutrophil biology and using not being focused really on inflammation, even if we use disease models. So what about neutrophils that go into the tissues at the end of the lifespan? So this is also published, so I will go quickly through this. So to start asking about the biology of the cells in tissues, and particularly how long they will live in those tissues, how long they would reside in those tissues. We built these mice, which is basically like the ketchup mouse, where you use the LY6 gene to introduce a Cree to allow recombination, but in this case is tamoxifen inducible, so it allows us to hit a specific time point and look for the wave of neutrophils as it moves from the blood into the tissues, and then it disappears. So this allows us, so this is just the validation that the animal is working specific. So this allowed us to design this experiment where we hit with tamoxifen, and then for the next 7 days, every day we track for the cells in the different tissue compartments, including the blood, and we use mathematical modeling and use the decay of the frequency of the number of neutrophils in the different compartments to estimate the half-life of the cells. So a lot of work compressed into this figure, but basically with the information that we wanted, which is the lifetime of the neutrophils being anywhere from 8 hours in the liver to almost 20 hours in the skin. In the bone marrow is 20 hours, but this is a bit confounded with the resident population, the maturing population there, so I'm not making an emphasis on this particular sort of. So bottom line is that the cells go into the tissues, we find them there, we published this already, but now we know that they have a residence time that is significant, you know, anything, it's a good number of hours. So even if they're not transcriptionally very potent cells and they will eventually disappear, this is still, particularly in certain tissues, a good number of the cells in high enough numbers that if they're producing any product or factor, they would be able to influence the physiology of that specific tissue. And this is what we wanted to, that was the intention of this experiment, to see if there was a rationale to study the biology of neutrophils in healthy tissues. This is always in healthy conditions. So again, it's published, so we'll go quickly through the different assays that we use to not only look for the presence of neutrophils, but also very much inspired by the work of many in macrophages, to look whether, since they have a dwell time of a good number of hours, whether they would acquire specific phenotypes that is specific for different tissues. And this is why we use cytoph analysis, which I'm not showing here, but also transcriptomic analysis. In this case, single cell transcriptomics. This is the neutrophil population in this case, these 4 tissues: bone marrow, blood, spleen, and lungs. And as you can see here, we find a number of clusters. When we transform these clusters into the tissue of residence where we take them from, what we found is that clusters, for the most part, match the tissue where we extracted them from. So the blood forms a cluster, the lung forms a cluster, and the spleen and the bone marrow, for the most part, overlap, which perhaps is not surprising within the spleen as a granulopoietic organ. And of course, with these, we can look at specific products that are produced in specific places, which is very informative, because it will allow us to look into potential dedicated functions of those cells in the different tissues. So cyclin precursors are here in this subcluster here. Early granule proteins that start being synthesized already at the early myeloid progenitor stages. More late stage different granule proteins synthesized later on in this subcluster here. Transcription factors specific for neutrophils, that specification that occur at this stage. Cytokines related to inflammation that appear in what we think are the late stages of pulmonary neutrophils, probably these mature neutrophils that have not yet been released, but also appear in the lung and in the blood. And then the specific cytokines or enzymes that are more associated with late stage. Once they leave the granulopoietic organ and they go into peripheral tissues, they acquire new profiles, and this is particularly interesting. So we combine the single cell features, which do not give us a lot of depth, even if they're very nice to look for specific subclusters. So we did bulk sequencing from, in this case, also the intestine and the skin, and we find this pattern that is also tissue specific. Many of these we could validate with the single cell data. There's a number of genes or the neutrophils from the skin and intestine seem to have actually the highest number of specific genes. And this is the validation against the single cell data, basically saying that what we predicted, or the bulk and the single cell data, go hand in hand. We also did, together with Irina Udalova in Cambridge, analysis of the chromatin using a TAC-seq, basically to look for more stable or more nuclear steady changes that occur on the neutrophils that go into specific tissues. Again, this is done only in these 4 tissues. And this, what we found here, again, is specific peaks, areas of the chromatin that are differentially open in different tissues. And this, again, matches quite well. And this is an example just showing that the signature in the lung with this tag sequencing matches quite well the signature that we expected from the single cell sequencing. So we put this together, and again, this was the idea to give a profiling, but now use this genetic information. I'm sorry, I have this. Sorry, the phone. So we put all this genetic information together and do a pathway analysis to see what the data would predict. And what we find was quite interesting, I think. Some confirming actually that bone marrow and blood neutrophils have a strong, consistent profile of what we call immune response and chemotaxis migration. This is interesting because these are the cells that basically 99.9% of the studies have been focused on. Blood or bone marrow neutrophils, either in steady state or during inflammation, have the canonical response. But what about other tissues? That's when other non-canonical functions appear. For example, angiogenesis or neurodevelopment in the lung or intestine, epithelial and connective tissue growth, profiling in the skin, B-cell homeostasis in the spleen, and so on and so forth. And this is just a handful of tissues that we could examine. So this is just a validation that some of these predicted functions were indeed translated into actual function, not only prediction. So we look for angiogenesis in the lung and in the intestine in control or in neutropenic mice using this MRP8-IDTR system, which is very efficient in depleting rapidly neutrophils. And what you can see here is in 2 different conditions, either sublethal irradiation or in very young animals that are still growing and where there's a lot of cell proliferation. And we look here for the percent of proliferating endothelial cells. So this in black would be the normal rate of angiogenesis or endothelial proliferation. And look how this goes really down in animals that have been depleted of neutrophils. And something similar shows up in the intestine. So no difference in the adult, just to make this point, but we find difference once we challenge the system. And this is specific for these 2 organs where we don't find the signature or this response in the spleen or the bone marrow, which were tissues where we did not find this angiogenic profile. So there really seems to be a good correlate between this initial omic analysis and what we found functionally, but it also opens the idea that there's a lot of biology to be understood of these cells in different tissues. So the lung has been a little bit our workhorse for several reasons. It's an organ that is a little bit more specific, if you will, than the bone marrow or the spleen, but at the same time it has a lot of neutrophils, so we can do some genetic analysis there rather than in the skin or the intestine, but there are very few of them. So focusing on the lung, which is a tissue where we're still actively working, one thing that got our attention is that the distribution of neutrophils there is not random, but rather around these CXCL12 expressing vessels. We suggested that they might be, like for macrophages and many other immune cells, some sort of niches or preferred areas where these cells go and possibly acquire their transcriptional profile and their function. So this is a quantification that the proximity is not random, but really is intentional in some way, and that the cells are in close contact with this with the tip of these vascular structures. And we used this screening using the signature gene expression signature of lung neutrophils to look for specific factors or signals that might be driving this profile. And we found CXCR4 one of the most relevant signals in driving this specification, which matches well with the presence of CXCL12, which is the ligand for CXCL4 in this particular niche-like structure. So really, this niche seems to be feeding on this particular network, and now we're trying to understand what is the molecular imprinting of the molecular regulation intrinsic to the neutrophil that goes into the lung to acquire this specific function. Always with the intention of understanding the function, being able to manipulate, and perhaps be able to interfere or manipulate the cells for some sort of benefit. So putting this a little bit together, again, emphasizing that there's this unidimensional view of neutrophils until not too long ago, where the cells are mostly have this exterminator kind of profile where they take care of infections, but they can also cause cell inflammation. But now we know from the works of many, including Dimitri, that they have a very strong prevalent roles, different, but a strong presence in repair tolerance and in tumors. And then we propose that this additional biology where the cell fits many tissues, and that the tissues allow acquisition of new functions that in turn contribute to these key roles in ways that we still don't understand, that we're certainly interested in understanding. So these different ideas, and this is what we're moving. For example, what about the skin? What is it that they're doing there? Are they helping build something? Hopefully I can share something along these lines next year. Functions to support hematopoietic cells in the spleen or the bone marrow, for example, neural reinforcement in the intestine, and so on and so forth. So really, I think exciting work to be done in the coming years. So stepping back, and with the idea of the blood being really a source of heterogeneity, this is circadian regulation that I mentioned, and feeding the tissues, we've been interested in playing with the circadian clock therapeutically. And what Alejandra Aroca and the lab is doing is just trying to hit these receptors, a way to prevent these physiological agents, which is playing with a number of new agonists against CXCR4. But anyway, that was not the point I wanted to make. The point I wanted to make is when we do analysis of the blood neutrophils from these animals treated with one of the agonists, or even in control situations, what we find is quite a varied response, transcriptional states of the cells. And this is something that is puzzles me a lot. You know, how can you have the same cell type released at the same time in the same conditions and featuring this wide range of parents? That's by looking at transcription. But if you look, as in my background has a lot of intraviral imaging looking at the cells, what about when you look into an inflammatory reaction where you see cells in action? And this is an example from a paper that we published a few years ago. And you see the same situation, the same setting, the same vessel, everything at the same time. Yet you see cells behaving differently. You know, it's subtle, but it probably matches this heterogeneity that you find at the transcriptional single cell transcriptional profiling. You have cells that are rolling, cells that are polarized, polarized by looking at the red distribution of selecting, cells that trap platelets, cells that move in one direction against the flow. You see all sort of things, minor differences, but they're there. The behavior is far from homogeneous. So we became even more aware of this when we studied this a few years ago. We proposed that the interaction with platelets in the circulation was an activating a triggering signal during inflammatory reactions. And when we were doing the studies, one thing we started realizing is that looking at the cells in 2D, like we usually look at them in intraoral microscopy, was not perhaps the right way to look at the cells, that perhaps it would be more useful to think of the cells in 3 dimensions. So you see the cells here distributing inside a vessel, exposing this PSGL1 enriched domain, which is what they use to as a bait for platelets, if you will, and this is what triggers signaling to the cell to start doing whatever they do during inflammation. But look at this, we transform this image in 2 dimensions into this image inside the vessel. So quite different if you think about it. And it also makes us aware that we know just a tiny fraction of things, perhaps in part because we're not looking at them in the right way. So this is the idea that I wanted to share. And this is what we are putting together now. And the idea is that we see things often in 1 or 2 dimensions. And what we've learned with this omic revolution, the single cell transcriptomics revolution, and all these new technologies, is that looking at cells in multiple dimensions has its benefits. Even if we don't know what a specific gene or factor does, the fact that it's there and contributes to define a cell is useful. So a lot of the effort has gone from going to genes or proteins, for example, when you use CyTOF, into a biological output, which could be cell identity or cell state, right? But when we do imaging and we look at fast reactions like the ones that take place in acute inflammation, basically you cannot take and do a transcriptome every second. It's not technically possible, and it would be too expensive anyway. So what we realized by with these images that we've been playing for a long time, is that there's this intermediate type of level of information that is captured probably by the cellular behaviors that I mentioned, the cell that is rolling, the cell that is attached, and polarized, doing one thing or do the other. They, in a way, are integrating signals that are coming from the environment or from inside the cell, and they behave. The behavior is a result of this network of interactions. And at the same time, these networks of interactions that hit, for example, this particular behavior, which could be the speed that the cell moves with, has an impact into the final biological output, which could be, again, cell identity or cell state, whether the cell will participate in an inflammatory reaction or not. So the question is, could we use the cellular behaviors to define, with good enough resolution, something similar to what we have typically been doing for the last 5 to 10 years by looking at single cell transcriptional profiles? Can we do behavioral profiling of the cells and learn something new from this? So again, this is an idea. This would be what defines this single cell omic transcriptomic, for example, which is what we've been using, but we cannot use this for acute reactions. So can we use behavioral profiling? So this is an example from experiments that Miguel Palomino, is now postdoc in the lab, did when he was with Santiago Gonzalez in Switzerland. And this is an infection of the trachea where you have in red dendritic cells and in green neutrophils. And this is what the raw movie looks like, cells moving around or not moving with different sizes and then just doing what they do. This is 3D, and just showing this in 2D. Now we can play this in a little bit more of a 3D kind of thing. So we can capture all this information of cells moving and volumes and shapes and everything. And you can put this together, and this is what this looks like. So this is the same thing, but now you can see the things in a different way, in a measurable way. Let's put it this way, where you can measure volumes and the tracks of the cells and the size and all sort of things that you can imagine, that you can extract by analyzing this. So what we realized is that we could describe these behaviors. Now we can extract a lot of information from this. We can extract, in this particular experiment, up to 118 parameters, many of which we were not interested in. but a number of, not interested in the way that they were highly redundant. So we cleaned this up a little bit, and we come up with 31 parameters that describe features of each individual cell here. And what we realize is that the more parameters that we apply to our analysis, and the Disney kind of idea, you know, just with similar maps, the more resolution that we get from this, you know, open space where each cell is its own. Basically, this would be your typical plot in one dimension that you would find in imaging analysis. As you refine this, adding more and more parameters, then you come up with unique maps. Now, we built this map. In this particular case, we thought information of what was a dendritic cell and what was a neutrophil, because we wanted to train the system so that we could now ask how, now that we see that there are more or less two clusters, two groups of cells, how well are we doing at identifying the most obvious biological difference between the cells, one being dendritic cells, the other one being neutrophils? Can we, is the system able to identify this particular and important biological feature, which is cell identity, by just measuring these plots here? And this is what you come up with. This is just a heat map showing what I showed you before, just put in numbers. So this is 31 parameters and the classification that you see here. And what you find here is that the clustering that you get by using, you know, your SERAT single cell analysis is almost identical to the one that you obtain. Sorry, this is from using the imaging analysis that I just showed. And this is by putting the tag, okay, this was a neutrophil, this was a dendritic cell, because we knew this, even if we didn't use this information to build this. And what we found is a very strong match, basically over 99% of being able to identify one cell type over the other, just looking at the behavior of the cells. So this is a way to map this in a different way. And this is very mathematical. This is a little bit crazy, but I find this very informative once we're collaborating with mathematicians to be able to digest this. And I like this. This is your typical person correlation map in a network form, where you have each parameter as a dot. The bigger the parameter, the better that it explains, in this case, whether it was a neutrophil or a dendritic cell. And with this sort of map, you can see all the relations, positive or negative, between the different cells. So this tells you how good was your parameter acquisition. The more cells that are clustered in a specific place, the more redundancy. The bigger they are, the more that they explain. The smaller they are, the less that they explain about a specific feature. So really, ideally, you would want to have a lot of mid-size balls or bubbles distributed along the space. So this particular data set or acquisition in this particular context was not perfect by far, but it was good enough that it allowed us to identify the different cell states. And this is a way to represent this, or just to give a message, is that the more variables that you acquire, in this case by using imaging, even if you don't know anything about the system, the more that you start learning about the biological properties of the system. In this case, this classification index, yes, was it a neutrophil or was it a dendritic cell? So that's the biology of this particular experiment. So this is cool because now you can use these maps and use your typical heat maps to see specific patterns that you might be interested in, whether, you know, the changes in speed or the duration of the track, the shape of the cell. You can see that the dendritic cells, which are up here, they have a gradient on this particular shape of the cell. Acceleration rates, track display, anything. It's very easy. The things that you might think this is ridiculous, it doesn't matter. It's ridiculous, but it helps you classify the cells. And in some cases, it might become handy. For example, you might be wanting to ask, you separate the clusters and say, okay, for neutrophils, what is the relation of the distance to the dendritic cell to the behavior of the cell? So the closer they are, is there something happening to those cells that I might be interested in? So in this case, you can use a Pearson correlation plot or look just visually and realize that the closer they are to the dendritic cell, the slower that they move. This might be predictable, but this approach allows us to capture this. So we just played with this system. We thought it was a lot of fun, so we just started playing. This is a model of ischemia reperfusion in the skin, and here we analyze a lot of cells and generate individual time points, so up to almost 50,000 time points that we analyze one by one using, of course, computers. And this behavior of the green cells, neutrophils, accumulated in the ischemic area, but not in the non-ischemic area. you capture with these 3D plots, which is what we use to build our information data set. And you see a lot of red and gray, and this is the cells behaving there. We capture this information and we build these plots. Same thing as before. This is the plot that we get by just looking at the behavior of the cells. And this is, we know which cell is a macrophage, which cell is a dendritic cell, and which cell is a neutrophil in this particular context. And we find that there's quite a good match. And the only mismatch to some extent is the incapacity to discriminate a subcluster of dendritic cells from macrophages. Now the question is it our system that is failing, or is it that the biology of those cells based on behavior, really it doesn't matter what the identity of the cell is, the cells are behaving the same way. So that's a question, you know, to think about. And one last example is from the skin. This is a laser burn injury, the typical swarming reaction of neutrophils, where you see cells going to the, you know, we burn with the laser, and then neutrophils start coming like crazy. So this is the classical swarming images, and then dendritic cells will form a plaque around this neutrophil cluster swarm. And this is how we see this in a data collecting way, with volumes and movements and so on. So we take these, we build our new Disney plots, and we again are able to discriminate quite nicely between dendritic cells and neutrophils. This is our predicted from the behavioral profiling, and this is the actual based on the expression of the reporter. So there's a very good match. You can find to what appear to be two different behaviors of neutrophils, and then you can take neutrophils, for example, from dendritic cells, and look for specific patterns that you might be interested in. So for example, the distance to the injury is as small as here. So this is where the injury is caused, and you can see patterns that appear in neutrophils. So the behavior of the cell is very much conditioned by the distance to the wound in the case of neutrophils, but not so much in the case of the dendritic cells. And this is one example of where you can look at here. Okay, so in those cases, we know about the identity of the cell. But the challenge is, what if you don't know about the identity of the cell? You don't know anything about the cell. And this was what we did here. We took the vessels, so this is going back to our neutrophils in vessels. And now we start looking, okay, they behave differently. What can we learn by doing the same type of analysis? That a cell behaves one way or the other, what type of information are we collecting from this analysis? Here, we don't have any reporter, anything to tell us anything, but we have at least these images. So same thing, we collect the images from inflamed vessel. This is the cremaster muscle, so a venule, an example, and this we induce inflammation with TNF-alpha. So we transform all these movies, there's a large number of them, into this heat map with thousands of cells and temporal cells at different time points, and in this case with 73 parameters that we could score. So this is a map. It's a little bit crazy, but this is the information that you can transform into the same sort of plot as I showed you before. Correlation networks, they start looking better. So more of these bubbles, medium size, separated from each other to explain the behavior. And this probably is because we have more or a better temporal and spatial resolution in these experiments that we had before. And this is what comes out from this analysis. What comes out using your UMAP sort of transformation into 2 dimensions is I like to see a bird here. So this is 3 clusters, this main cluster in pink, this behavior, what we call 3, and behavior 1. So this is just tags. You know, we see different groups, clusters, when we say, okay, there's 3 things at least going on here. But now we can use statistics and say, okay, what discriminates, what is different between the different clusters from the data set that we have, the distance to the wall, how much they move, the size of the cells, the shape of the cells, whether they're prolate or they're oblate, how much they're standing on the endothelium. So we can plot this to compare all these populations. And the reason this is just to give a visual idea of what we mean by this. So we take all these parameters. What does this mean? So this behavior here, this behavior 1, is consists of cells that are sort of round. They're prolate, so they have they kind of polished shape, but is standing on the endothelium and they don't move. So they just sessile. Behavior 2 is cells that move a lot. So there's your typical crawling neutrophil inside vessels that move, you know, on the vasculature, doing the crawling, the typical crawling that people have described. And then this behavior 3, both 1 and 3 were not reported as far as I know in the literature. And this behavior 3, where the cell is very big, is sessile, it doesn't move. And you will see in a second that it also has other specific properties. One thing that we learned, from example, by looking at this behavior 2 is that these cells were moving the specific high standard deviation in movement, in many parameters related to movement, which suggested that each individual cell changed a lot the shape. So when we look more carefully in the reconstruction of these cells, what you find is that the cells are moving sort of like an accordion. So the cell goes from round and then it stretches and then rounds up again and then it stretches again. So they don't crawl like we thought of an amoeboid movement, but rather the crawling is sort of like a, I don't know what the name of this worm is, but like an accordion that is advancing and then contracting, advancing and contracting. So the side view and the top view. And the other thing that made this particularly useful for us is that we generated this map by using 2 conditions. One, in a wild-type animal, we know the animal is susceptible to pathogenic inflammation, and this is the distribution of the cells. So most of the cells in a wild-type condition will go to cluster 3, to group 3, and to group 2. Now, if we repeat the experiment in animals that have been depleted with platelets, we don't see basically any cells in this behavior 3 group, but they're mostly in this behavior group 1, these cells that are like this, and in behavior group 2. So behavior 2 is sort of common to the 2 groups. So now this is interesting, as I mentioned, because the control animal is susceptible to inflammation. The platelet depleted animals are strongly protected from inflammation. It's something that we know for a few years now from this study that I mentioned before. So this behavior 3 that is quite specific to the control group that is susceptible to inflammation. So we associate this behavior to inflammation. So to animals that have inflammation, have quite exclusively this type of behavior. And one thing things that we find that we think are particularly interesting is that these cells, they're not only large and flat on the endothelium, but they also have a footprint kind of the endothelium, and they're actually semi-wrapped by endothelial projections on the cell. What this suggests, but we don't know yet, but what this suggests is that the cells, if they get activated, they're in closest contact with endothelium, and they're more likely to be able to induce some sort of injury into the vessel wall. So that's the way that we see at this, but we don't have actual proof of this. Now, again, you can play with this and you can add a little bit more refining. So cluster 2, you can divide into other clusters. So you have 3 clusters that would be the behavior 1 that I mentioned before in the inflammation protected group, behavior 3 in the inflammation prone group, and then this intermediate state that happens to, you can classify it into 5 groups. And you can now, because we have information from each single cell, draw lines and ask if there's specific jumps between one behavior and another. Whether this is, a cell behaving one way, will they stay always this way, or you can transit between different behaviors. And this is where we quantify here and propose this sort of model, that the cells go from a non-inflammatory sort of sessile inert form into a highly migratory that can transition into inflammatory, as you can see here. How am I doing with time? I'm almost done, Jennifer.
Oh, good. Yeah, we're at time, but yeah, so finish up, I guess, and maybe we can do a couple questions.
Okay, I'll just wrap up quickly. So anyway, with this, using this criteria, now we have a criteria to define what we think are pathogenic states. So we think about neutrophils always, but there's a pathogenic state and a non-pathogenic or protective state, if you will. And we can use this to now ask in a number of mutants whether they would favor one or the other type of behavior. With the idea is which is this behavior deterministic? Are there specific genes that drive one or the other behavior? And basically, I'm just summarizing here 6 years of work, mostly of Georgiana Kranichuk in the lab, into one slide, into one panel, just to show this map that we will be with 24 different mutants and looking for specific behaviors to basically come up with behaviors that were protective. So if we knocked out a gene, then the cells adopted this protective state. And we were interested in this, obviously, to look for ways to prevent inflammation by preventing the activation of neutrophils. So anyway, we identified FGR, which is SAR kinase, and we repeated the experiments, these behavioral maps in the FGR knockout or using an inhibitor, and found that, you know, as we predicted in our screening, they basically move away from this cluster 3, which is the pathogenic one. And this is just to show that indeed, when we now analyze these animals in 2 different models of acute inflammation, one is myocardial infarction, the other one is glomerulonephritis, so acute kidney injury, then there's almost complete protection from injury. So last slide, just to summarize, the idea is that there's a lot of granularity, a lot of diversity and non-canonical functions and ways to look at the cells that might be useful, that illuminates ways to understand really the biology of these cells. I'm particularly interested in this granularity that exists in the blood and possibly in tissues as well. And when we talk about biological noise, in some cases, I think this noise bears a lot of important and interesting information that we can use to learn more about the system and possibly for therapeutics as well. And this idea that there's this in blood, these transitional states in the context of inflammation. So this is the lab. And I just wanted to highlight a few people. Jose Maria, who is not here anymore, who was very much involved in the circadian studies together with Maria Casanova, who left a while ago and is now an independent group leader. And this is mostly also the work of Georgiana, Miguel, and John here, who have been working hardcore in this last part on the context of inflammation. But really everybody, Alejandra, everyone contributed a lot to this. So thanks so much. Sorry for the extra time. I always go too long, and I'll take any questions.
Thank you so much. That was a fantastic talk. We have a lot of questions. I think maybe we'll let people ask directly because some of the earlier questions, you know, might now be superseded by your fascinating recent work. I do have a question first from myself, which is, you know, this type 3 neutrophil is associated with pathogenesis, but what do you think the normal physiological role would be, or is it not normally present except under like these critical inflammatory situations? Yeah,
I guess, yeah, it's hard to tell because we don't see those cells transmigrating, which is what you would expect. The cells are on the way to transmigration. They you know, for a few minutes I will look at them and the transmigration is fast. The cells are just stay put. We don't see any. That probably will be a question from the reviewers, but initially the cells stay there. They're not going anywhere. And typically when they transmigrate, they have this very polarized and migratory phenotype. So we don't know if this might be a noise in the system, basically in the system that we're using with TNF-alpha. More likely some, you know, there's these patchouli monocytes that inspect endothelial cells and look for death, or, you know, remove excess lipids, who knows? And we don't know if these cells would be there just even at the beginning when we have not done anything in specific tissues that might be adopting this behavior. We don't know why there's this extreme behavioral phenotype, but certainly is there and is associated with. Unfortunately, we've only done this TNF-alpha model of inflammation, so it would be, I think, quite important to look at other tissues or you know, use septic shock, something like a really strong response, and see is this really is a default of the cell when it goes too far, but it's unable to transmigrate. So an open question, a good one, but an open question.
Great. So we have a request for our questionnaires, which is, can you use the raise your hand feature in the participants box, and then we can call on you. And let's see, Jennifer.
There we go. There's one.
Sanya,
go ahead.
Hi, I'm Sanya Rangelis from University of Virginia. That was a great talk. Thank you so much.
Thank you.
So my question is related to the neutrophil aging phenotypes that you guys see. If I understood you correctly, the aging process is resulting in this sort of degranulation kind of a phenotype. And yet, in some of the models, you see that these aged neutrophils are better off at fending off pathogens, and they are more injurious, for example, in your IRI. So how are these degranulated neutrophils so good at inducing injury?
Yeah, that's a good point. So we use the CXCR4 mutant, which is not the best. It helps for certain things, but it's also because they are released prematurely from the bone marrow. So there's a bit of a disclaimer that there's some of these things we validated in a different system. The things that we don't understand, but the way that we think about this, so not everything matters. I'm not gonna say that my model is the best, held to the rest. No, there's things that don't always match. The one thing that would make things a bit consistent that didn't show here, but it's in the paper, is that aging is required for extravasation in the tissue. So if you block aging in the BMAL mutant, for example, then the entry into tissues is impaired. Now the question that we ask, but it's a different difficult question to answer, is if the retention inside the vessels, if part of the programming, and we saw this, when there's aging, the microvilli retract, integrins are exposed, and the cells attach better, and they presumably are able to go always in a steady state better to tissues to extravasate. If you block that, then you would have cells that would go into the vessel, but cannot quite complete the migration into the tissue. So what happens now if you're unable to clean up the cells properly? So first you would have more neutrophils inside the vessels, but also cells that eventually, I don't know if they would go into some sort of apoptosis, necrosis, or some sort of. So the preventing the transition is one thing, and the phenotype itself is another thing. You're absolutely right. So the CXCR4 are less potent at inducing injury in the lung model, but in the lung model, they don't need to extravasate to go anywhere. Compare, for example, with a myocardial infarction, in which they do need to go there, or an infection where we think is that they really are outside the vessels, in the tissues, and probing for things to eliminate before. So there's a lot of question marks. I cannot basically, there's a lot of points that we would predict that we haven't been able to look at to explain how the whole system works. But this is a particularly good point. Why does the CXCR4 have different phenotypes? And that's absolutely right. The only difference is one is a purely vascular model of intravascular inflammation, and the other one, the cells will need to go out of the vessel. So whether that's what explains the difference, that's the one answer that I can give right now.
I have a question that follows from this and from some of the chat questions. Do you think these intravascular neutrophils are promoting vascular leak?
If they're promoting?
Vascular leak? Vascular permeability?
Yeah, it's hard to tell. I don't think so. Back in the day when I was just looking for extravasation of dyes, dextran, of these sort of things, having an inflammatory reaction in vessels, for example, with TNF-alpha, was not sufficient. You really need to put some stress to the cells, or really induce an acute response. Like in the Trally model, you give an antibody that turns the cells a bit crazy, induces net release and a lot of ROS. In that context, I started seeing a little bit of leakage. For the most part, the endothelium seems to be quite resistant to just leakage, because you have inflamed cells transmigrating. There's all this body of literature of how a cell would transmigrate without leaving pores and opening into that seems to be quite controlled by the endothelium.
Well, we did have some really interesting questions in the chat, so maybe I'll ask a couple of them. A really interesting question from Tatiana Novobrantseva was, is neutrophil function affected by temperature elevation? How does a fever affect your different types of neutrophils?
That's a good question. I don't know. I mean, I'm sure I could pull out from PubMed, but the things that we haven't looked. One thing I can tell you related to fever, that's a good point. I always turn things around. I'm sorry, but rather than how they respond to fever, can neutrophils cause fever? And there's evidence actually that they can, because they produce prostaglandins. So that I can tell you, they will respond different to temperature. I have little doubts that they will, because they respond different to almost everything that you hit them with. But I'm sorry, I cannot be more specific, because it's not something that I have looked into.
Jennifer, do you have a favorite question to ask?
Yeah, so I mean, maybe also, Andreas, we could send you these questions for anyone who's jumped off, and if you might respond to them. We could put them on the website too, just because some folks have left. There are some good ones here. I mean, there's some sort of like logistical questions about collecting neutrophils, and if the time of the day matters, or clearly it matters, but should we be trying to enforce that it's collected at the same time?
Yeah, the general answer to that, yes, please do the experiment at the same time of the day if you don't want to go crazy. Yeah. Actually, my postdoc mentor published a paper in Nature a while ago based on one of these, someone in the lab was doing the experiments with the light zone or different times of the day, and they couldn't quite reproduce things. So Paul is a smart guy and say, hey, what's going on? And he came up with all this circadian regulation in that particular case of stem cells. So by all means, unless you want to make some groundbreaking discoveries that will take you a lot of time, just do the experiment at the same time of the day. Noon is the best day to collect cells in a mouse from the blood, because you have like 10 times more than what you have at nighttime.
So interesting.
Here's an interesting question. Do you think that the neutrophil plasticity or types that you've observed are derived from the bone marrow or are dictated by the tissue?
That's a very good question. One that we're interested in. Yeah, the ones that we found are the ones that one finds in cancer or in lupus or in a lot of things, or that go into the different tissues. That's a very good question, and it will take us a little bit to answer it, but we're gonna try to do some clonal tracing to answer that question. We don't know. It might be, there's some ideas around that, but it's not technically easy, and I hope that we'll be able to do something around that.
Would anybody like to ask a question in real time?
I see some questions here. I don't know if anyone prefers to.
Here we have. Go ahead.
Thank you so much. I'd like to know your opinion about the controversy and distinguishing between GMDSCs and neutrophils based on the tissue type, and what are the best possible ways to distinguish them, and also, are your neutrophils CD66B positive, and what other markers have you used to?
In mice, we never use CD60C, unfortunately. Right. So in humans, of course, CD60B is one not super specific, but a good marker for neutrophils together with CD16. So it's a tricky question, because Dimitris is in the audience. He should be answering this question, and this has been a lot of. It's just, I think, a matter of nomenclature. And MDSCs are just at least the granulocytic are just one of these shades of neutrophils, and I think everybody would agree to that. But it's one that is associated with specific diseases, so I think it's been useful to use the nomenclature. It's been abused sometimes, like, how this mark is C11B, GR1 positive, there you go, MDSC, or yeah, MDSC and any other name that you want. That is not specific enough. But if you associate it with a disease, or you think of them as a state rather than a subset, and I think there's a beautiful review that just came out yesterday or 2 days ago in Immunity from Miriam Mera. That would be very useful for you to look at. Because they make the point that these are states, they're specific ways to define them, but they're not universal. So it really, the point is use function as a way to define cells. This is what MDSC does, by giving a new name, it makes it confusing. So talk about neutrophil states, if you will, or monocyte states. Because you could also talk about MDSC's macrophage, right? If they're suppressive in one way.
Yeah.
So that's my thought around that. It's been a useful concept, but nomenclature wise is a bit confusing because C11 BTR1 positive cells are neutrophils.
Thank you so much. Thank you.
Thank you.
We have a question from Naharika Jain.
Great talk, Andres. I was wondering the different stages that you capture from the images. Is this stages or different features that you capture from the images which defines different clusters of neutrophils? Are these a continuum, or these are completely different stages that don't mix up? They don't move from something like migratory to stationary stage or a stationary to a much bigger stage. And how does that define the cellular interaction profile of these different clusters?
Yeah, so I saw, maybe I went too quickly at the end. I saw one slide showing that we could measure the transitions from one stage to the other. So they're not locked transitions. So from, let's say, B1 to B2, or B2 to B3, or B3 to B2, we see quite often like 20%, 25%, 1/4 of the cells would jump from one to the other. The transition between 1 and 3, so they really sessile forms that are quite different, that's very rare that we measure. We measure just very few of those. So I think it's a continuum, but it's a continuum that it is not everything goes into everything. It's a continuum where 1 goes into 2, goes into 3. And this is interesting because in the genetic models that we use, the idea is one could try to define what are the regulators between one transition and the other transition. If you want to, for example, go into one state where you want to, have a robust response, or if you want to block it. So how much of this is deterministic on the cell having a specific factors or proteins driving this out? The point is that we can use this tool now to ask this question. I mean, it's the only way, I think, so far to look at functional states in vivo. And this and other questions, hopefully, because we have stored information from each cell. We can ask a lot of these questions that you're asking.
Yeah, because I was wondering, unlike single cell data where we have parameters like we can define RNA velocity and we can capture it on a pseudo time, are there techniques out there where we can do a pseudo time kind of thing on imaging data sets? Yeah,
well, we did pseudo time in that slide that I showed you, except there it really is the same cell. So you don't have to infer it by, for example, the splicing region. You can, like velocity does, you can just go, okay, this cell is the same cell that I saw, and it goes from one state to the other. But the idea is the same. I mean, a lot of the ideas that we use are feeding from the single cell technologies, that tools that have been developed. The answer is yes, you can do it even better than you do it with single cell transcriptomics.
And if may I ask one more question. So targeting CXCR4 and in different niche, when we are talking about the profile in gut or the profile in lung, which seems to be very, very different based on where neutrophils are. How much do you think targeting CXCR4 for any phenotype is a viable option? How does that affect other myeloid cell functions as well?
And you mean, sorry, I missed a little bit. You mean in our particular model, if it's affecting our myeloid functions, or is the cells not having, the neutrophils not having CXCR4 that are affecting other cells? I'm not sure if I get your question.
So no, those two questions are two different aspects. So like in your model, when you are deleting CXCR4 or blocking that signaling, how does neutrophil affect other myeloid functions? Because
you mean the neutrophil that does not have CXCR4 having an effect on other myeloid cells? No, we have not looked into that specifically. I'm trying to think if we have any. We haven't profiled, for example, we did profile actually cytokine production, things that might be affecting other cell types, but I don't have any good answer to that. So sorry, I won't be able to be more specific. We haven't done co-cultures. We haven't this effects in general physiology, but not one that we have, as I know, this cell is affecting this other cell, and therefore we haven't done those analyses yet.
Thanks, Andreas. Great talk.
Thank you.
Nice to hear you.
We have one more question in the chat from Dominique Rubinich. He wrote, I have observed that neutrophils can live longer when in contact with tumor cells. Do you think that their functions would be impaired?
When they're, yeah, no, not impaired. I think they would probably do something different. I think actually there are people who are using our mice, this reported the tamoxifen inducible model to measure the lifetimes, and then other groups like Michael Peter, people who have been doing quite a bit of work. So they live longer in part by the presence of the cytokine cocktail that you have there. And yes, they would not do more or less, they would do something different. And I think people have shown immunosuppression and angiogenesis as to clear new things that they do.
Well, we had many comments in the chat about what a wonderful talk this was, including Lisa Cousins, who thanks you for this presentation. And we really appreciate this fantastic body of work. I look forward to hearing more and seeing what you do when you move to Yale. Hopefully we can all get together and
yeah, that would be wonderful.
Yeah.
And I hope the myeloid, this seminar series will continue for a long time, and I will be happy, in the second or the third round or the fourth round or whenever, to come back again. This is very good.
So our next speaker will be Mickey De Palma, who will join us from Switzerland and tell us about his latest work, and that's in about a month. So please join us, everybody. And if you have questions for Andres, please send them to him directly and or through our website. And thank you again. It was fantastic.
Thanks so much.
Okay, take care.
Thanks, Andreas. Bye, guys.
Bye.
See you later.